Great to see everybody. Thank you very much for joining us. We have a mixed format. It's offline, online. So maybe you cannot see some of us very well, but it's okay. First of all, thank everybody for joining. And secondly, have introduction of a jury and the candidate jury members. We have quite a big committee, the jury committee today. And how do I change slides here, Lena? Okay, so I am the first one because I guess I'm the oldest on the list. My name is Philip Kaitovich. I graduated from Moscow State University in 1995. I'm a molecular biologist by education. I worked in United States, in Max Planck, and in Germany, and in China for a total about 20 years, and I returned back to Skoltech, to Moscow in 2014. So I'm now here at Skoltech, head of Neuro Center. And the next one is, to remember, is Professor Sergei Dmitriev, who's a professor at Belozerski Institute. He actually graduated from the same department as me, from the Moscow State University, from the molecular biology, and but a few years later, and he worked abroad, and now he works at the Physico-Chemical Institute and has really a lot of publications and a lot of work done on the properties of RNA and other molecules. The next one is Professor Dmitrii Ivankov. Dmitrii is also here at Skoltech. He is a physicist by education, not a biologist. He graduated from the Physico-Technical University of Moscow, the best school for this direction at the time. He worked quite a lot abroad in Germany, in Spain, in Fedor Kondrashov lab, if I'm not mistaken, in Austria, and he joined Skoltech about 4 years ago and works here on the protein biology. The next one is Professor Gabriel Leprevier, from working now in Düsseldorf, Germany. I believe Gabriel is from France originally, and he worked a lot abroad, of course, as everybody does these days, and his main focus is on the tumor and tumor adaptation, brain cancers, and glioblastomas, I believe, at the moment. Next one is Professor Pervushin. Professor Pervushin is associate professor here at Skoltech again. He has several degrees, actually. He has degrees in mathematics and chemistry, and he also has a degree in the neuroscience, as far as I know. He's a distinguished professor at Skoltech, working on many topics of computational biology. He worked, again, a lot abroad, of course, as probably everybody on this committee, but maybe even more places than other people. And he joined Skoltech already quite a while ago, I think for the last 6 years, Dmitrii working at Skoltech, if I'm not mistaken, maybe 7 years. The next one is Professor Leonid Peshkin. Leonid Peshkin is working currently at Harvard Medical School. He has a very spectacular track record, of course, he got his master in applied mathematics from Weizmann, but then he moved to the United States, worked in Brown University, in MIT, in Harvard, so really the top places and has a lot of publications on computational biology on different topics. And the last but not least, from the jury is Carlos Sebastian, who is senior professor at the University of Barcelona. He received his PhD in immunology in University of Barcelona, then worked abroad, and his focus also on metabolic regulation and tumorigenesis, and with kind of things. Then supervisors and co-supervisors. So there are two supervisors here, one from Skoltech and another one from Ben-Gurion University. The supervisor from Skoltech is Professor Ekaterina Krameeva. Ekaterina is bright computational biologist, published many papers already on different aspects of analysis of the chromatin architecture, gene expression evolution, and diseases, and now switching her attention to study of aging as well. We're very lucky to have Ekaterina working here in Skoltech with us. She became a professor in 2019 here. And co-supervisor is Deborah Teuber, associate professor at Ben-Gurion University of the Negev. And Deborah received her PhD at Hebrew University, the best school as far as I know in Israel. Then she did her PhD in Harvard Medical School, certainly a place which needs no introduction, and rejoined Ben-Gurion University in Negev, or joined, I should say, at 2014 as a senior lecturer. She has many awards, and her research interest is molecular mechanism of aging and aging-related diseases, and we believe today's topic of thesis defense will be closely related to this issue. And finally, the PhD candidate, Dmitrii Smirnov. He doesn't have such long track record yet, so we will see what he's done in the past 4 years. He received his master and bachelor from applied mathematics and physics from the Moscow Institute of Physics and Technology, the same as Professor Ivankov. So this is again the best school for physics and mathematics at that time, and now I believe as well. So we expect a very bright defense. And with that, let us go to the candidate presentation, I believe.
Thank you for the introduction. Indeed, I'm Dmitrii. And first of all, I would like to thank everyone for being here today. So today I would like to present my results obtained during the all years of my PhD studies regarding the role of sirtuin-6 in the regulation of gene expression, metabolism, and aging. My work focuses on sirtuin-6 functions in the brain. Sirtuin-6 is a member of the sirtuin family that consists of 7 proteins in mammals that consume NAD+ to provide their main density, to provide their main functions. Main function, histone deacetylation. However, all these sirtuins, they are localized in different cell compartments and therefore have slightly different functions. I'm studying sirtuin-6, that is nuclear protein. And apparently it's the only one member of the family that can combine 2 enzymatic functions. As I already mentioned, histone deacetylation to influence chromatin state. And another function of him is the mono-ADP ribosylation to provide some post-translational modifications of other proteins. And because of these two main its functions, C26 is implicated in many intracellular pathways, has many intracellular functions. For example, it was shown to be the regulator of the gene expression. Of course, it acts as a chromatin remodeler because of the histone acetylation abilities. It also facilitates DNA repair, acting, for example, as a sensor, as a sitting at the broken site in the DNA. It has clear roles in cellular metabolism. For example, it regulates glycolysis and also acts as a regulator of lipid metabolism in liver. And due to all these functions, all the intracellular pathways that C26 involved in, it's also implicated in the aging and age-related diseases with the neuroprotective functions. C26 can be considered as a marker of aging. It was shown that in many tissues and organs, C26 levels decreased during aging. And as I said, it's in many tissues and cells and organs, and including the brain, where it also have decreased levels in some neurodegenerative diseases like Alzheimer's disease. And in opposite, overexpression of C26 can extend lifespan in mice. Originally, it was shown for female mice, and quite later it was shown for both males and females. So C26 targeted system can be was quite used in the longevity research. So yes, as I said, they were quite used in the longevity and aging research. And I would like to say a couple of words about C26 deficient models. If we block completely expression of the C26 in mouse, this mouse will be dead in approximately 4 weeks, and it will develop the progeroid-like phenotype. It was for full body knockout. If we talk about some tissue-specific models, for example, in heart, such mice will have some events like cardiac hypertrophy, and it's also C26 deficiency in heart associated with the heart failure and cardiomyocyte senescence. In liver, depletion of C26 was associated with increased glycolysis, alteration in lipid metabolism characterized by an increased level of triacylglycerides. No less impressive results were obtained for kidney and for C26 deficient models with kidney. In such models, it was shown the podocyte senescence, and such kidney-specific C26 deficient model were also associated with chronic kidney disease. And last but not least is the ablation of C26 in the brain. In the brain, it has one of the most striking phenotypes, resulting in the neurodegeneration and somatic growth retardation. Also, the ablation of C26 in the brain, it enhances post-translational modification of tau protein, hyperacetylation and hyperphosphorylation, increasing its toxicity. And also mice lacking C26 in the brain, it develops cognitive impairments that become more dramatic closer to the 4th month of age. So the importance of C26 for the brain, it's clear. However, the mechanisms through C26 can function in the brain especially mechanisms lying in the transcriptional and or metabolism level, they're still unclear. So in my work, I focuses on the bioinformatic analysis of the changes, of the sirtuin-6 deficiency-induced changes in the brain to clarify the role of sirtuin-6 in the cellular homeostasis using the brain-specific sirtuin-6 knockout models. First, we aim to, in my work, I aim to identify the changes, identify the signatures, transcription and metabolomic signatures of the sirtuin-6, brain-specific sirtuin-6 knockout mouse brain. And we also aim to investigate the mechanisms behind these changes, behind this SIRT6 dependent regulation, and connect these changes with the normal and pathological aging brain. To achieve this goal, my colleague from BGU generated a set of omics experiments using wild type and sirtuin-6 knockout data. For example, for transcriptomic and lipidomic data, they utilized brains obtained from mice with, as I said, from wild type and sirtuin-6 knockout brains that was generated using nesting-crate technology and nesting-crate system. And also they provided, they performed an experiment, metabolomic experiment with mouse embryonic stem cells, again, wild type and sirtuin-6 knockout groups, so 6 knockout conditions. I did bioinformatics analysis of this, of the obtained profiles. And first of all, we started with the analysis of lipidomics data, and the analysis of lipidomics is still a challenge. It's still challenging because of the choice. You need to choose the correct sets of parameters for some peak picking software, peak alignment algorithms, and you also need to the annotation is also tricky for the lipidomics. And to address all these issues, we developed a pipeline focused on mostly on LCMS lipidomics data, LCMS lipidomics data. However, it can be also applicable for metabolomics. In this pipeline, we implemented all the steps of the lipidomics analysis, starting from raw data processing and to the downstream analysis, including the normalization and statistical analysis of the obtained lipid abundances. As I said, we implemented for downstream analysis, we implemented some statistical methods, both univariate and multivariate methods, like PLSDA, SPLSDA, and the pipeline is written in R. It was published in Metabolized Journal. And we applied this pipeline for the analysis of mouse brain lipidomics profile derived from 3 wild type and 3 sirtuin-6 knockout brains. Many thanks to my colleagues, Dr. Shai Kaluski and Dr. Veronika Isinsky, who did the experiment. We applied our pipeline to the obtained spectra, lipidomic spectra, and while the annotation of the lipid species was more or less looks more or less fine, so we even obtain a typical grid-like structure for triglyceride species in the mass to charge to retention time plot. However, we found no significant difference between the conditions, difference in abundance between the conditions, mostly because we have a limited number of lipidomic samples, and we had an issue with unbalanced design. However, several lipid species they had quite high log-fold change values for the differences between knockout and wild type, but it was not still enough for the statistical significance. It wasn't enough to make some clear conclusions. Originally, we didn't include these results into the thesis, but since several questions were raised during the review of thesis regarding the pipeline, we decided to include this part as well. Shows that attempt of the analysis was made. And given the unconvincing results with lipidomics and encountered limitations in the analysis, we proceed with transcriptomic data and metabolomic data. Let's start with RNA-seq analysis. We analyzed 4 wild type and 4 brain-specific 6 knockout transcriptomic profiles. And this analysis revealed a clear difference. First of all, we did a PCA analysis to clarify how samples are similar to each other. Wild type group, 36 knockout group was clearly different from the wild type, while inside the groups, the samples were very similar to each other. And consistently, analysis of diff expression also revealed a high number of differentially expressed genes. So I think more than 10% of detected genes in total were differentially expressed between the conditions, confirming the crucial role of sirtuin-6 in the transcriptional regulation in the brain. Then we investigated these genes and found that most of the changes for downregulated genes are associated with mitochondria, some essential mitochondrial processes. They're related to mitochondrial organization or mitochondrial respiratory chain complexes. And given the results with this mitochondrial, with the differentially expressed genes associated with mitochondria, we speculated these mitochondrial-related changes could be also observed in the metabolomics data. So we analyzed metabolomics data from mouse embryonic stem cells. And again, the profiles were clearly different between wild type and sirtuin-6 knockout. We also, our analysis revealed a high number of differentially abundant features between the conditions, both upregulated and downregulated. And then we started to think if these changes are also somehow related to the, for example, Krebs cycle, or for example, to the mitochondria in general. So we classified these differentially abundant features into several groups according to their functions, and found that indeed for a lot of differentially abundant features, they're associated with, for example, Krebs cycle, where they're associated with oxidative phosphorylation and energy pathways in general. And most of these changes, they were co-directed in our data. So we observed downregulation of most of the features, most of the metabolites in the sirtuin-6 knockout samples. While, for example, in amino acid metabolism group of metabolites, the changes were rather in opposite direction, since we saw some upregulation of the related metabolites. Next, we return to the differentially expressed genes, mitochondria-related differentially expressed genes, and focused on the specific pathways they evolved in. First of all, we classified them according to their to the localization of the proteins they encode, and found that these proteins, the proteins they encode, they overrepresented in the mitochondrial inner membrane the springboard for ATP synthesis. Therefore, we decided to investigate the pathways, mitochondrial pathway, perform mitochondrial pathway analysis that reveal enriched 5 altered mitochondrial specific pathways that are altered between wild type and sirtuin-6 knockout, and 4 out of 5 of them were related to the oxidative phosphorylation and oxidative phosphorylation complexes. And what's more impressive, all of these changes observed for the genes related to oxidative phosphorylation complexes, these genes were downregulated. So we saw the decrease of expression of genes that are encode electron transport chain complexes, proteins, and only one of them is DHA that performs the conversion of succinate to fumarate. It was upregulated in knockout. However, all others were decreased, as I already said. And my colleague Ekaterina Yermenko, she was able to validate this effect experimentally using flow cytometry methods and several mitochondrial electron transport chain inhibitors like rotenone, peroxide, oligomycin. She showed that mitochondrial membrane potential that showed as an MFI, mean fluorescent intensity, it's decreased in sirtuin-6 knockout cells compared to wild types. In all the comparisons with electron transport chain complex inhibitors or even without, so the changes were very consistent. And she also showed the increased ROS production, reactive oxygen species, in sirtuin-6 knockout cells, as well as reduced mitochondrial content, also in sirtuin-6 knockout cells, highlighting the role of sirtuin-6 in the mitochondrial oxidative phosphorylation process and mitochondrial biogenesis. Okay, so what we know so far is that sirtuin-6 acts as a major transcriptional regulator in the mouse brain, and especially it's especially related to the genes encoded to genes relevant to mitochondria and mitochondrial oxidative phosphorylation, mitochondrial the functions that are relevant to the energy production. It's also, we saw it in transcriptomic and metabolomics data, but we still don't know through which mechanisms sirtuin-6 can accomplish its mitochondrial regulation. So we started to search for the possible candidates for the through which sirtuin-6 can alter the mitochondrial transcription and mitochondrial metabolism. And first of all, we focused on mitochondrial sirtuins, sirtuin-3, 4, and 5, that encode that localized in mitochondria, and they're involved in several essential mitochondrial processes like we already mentioned oxidative phosphorylation, fatty acid metabolism regulation, production of reactive oxygen species. We investigated the expression changes of these genes in our data set. 2 of them, sirtuin-3 and 4, downregulated in sirtuin-6 knockout, while sirtuin-5 changed insignificantly. Next, we validated these changes in the public RNA-seq dataset from the brain with many observations. And here we saw the correlation of sirtuin-6 expression with expression of all mitochondrial sirtuins with sirtuin-3, 4, and 5. All the correlations were significant. But since in our data we saw only the changes for sirtuin-3 and 4, then we proposed only sirtuin-3 and 4 as companions for the sirtuin-6 to regulate the mitochondrial transcription. and metabolism. Another possible axis of the mitochondrial regulation is it's related to the its cooperation with YY1. It was previously shown that YY1 and sirtuin-6 can form a complex to regulate many intracellular processes, for example, that are related to aging. And it was shown that YY1 can bind to the promoter of sirtuin-6, and it also can activate its promoter and enhance its protein levels. So YY1 can, actually it's also a chromatin remodeler. It can achieve the regulation of the chromatin, impact the chromatin through several ways. For example, it can cooperate with histone deacetylases like sirtuin-6 to repress gene transcription. It also can cooperate and can interact with histone acetyltransferases to activate the transcriptions, And another way, it can cooperate with several long non-coding RNAs. And it was shown that its cooperation, its interaction with long non-coding RNA has a crucial roles in pathological brain, and these interactions evolved in, for example, Alzheimer's disease, and what is more relevant to my thesis, into the glioblastoma disease. So together with my colleagues from the Barak Rotblat lab, we focused on YY1 roles in the pathological aging brain. And we found that the YY1 can regulate the expression of TP73 IS1 long non-coding RNA in the brain. And TP73 IS1 is it's a marker of medulloblastoma, both medulloblastoma and glioblastoma, very aggressive brain cancers. And one of them, glioblastoma, it's even more aggressive in older people, connecting YY1 and brain aging. So we also show that TP73 IS1 can be is increased in normal aging brain and pathological aging brain. And my colleague from Barak Rotblat lab, Belmezor, she performed several experiments to confirm the regulation, YY1 dependent regulation of TP73 IS1. So with knockdown of YY1, she measured an expression, and she found that depletion of YY1 can downregulate the expression of this long non-coding RNA TP73 IS1. And she also showed that YY1 overexpression can increase the activity of TP73 IS1 promoter. So we published this story in Aging Journal. And given the role of YY1 in the pathological brain aging and normal brain aging, and given the we know that it can interact with sirtuin-6, we wondered if YY1 is also can cooperate with sirtuin-6 to regulate mitochondrial targets in our, for example, in our data and in general. First of all, we analyzed two publicly available ChIP-seq data sets for the YY1 and for the sirtuin-6, and found that in both of them, YY1 and sirtuin-6, they bind to the promoters of the mitochondrial related genes, of the genes that encode proteins localized in mitochondria, specifically in mitochondrial inner membrane and the mitochondrial protein containing complex. And then we superimposed this these genes from these mitochondrial targets of YY1 and CD6 from the ChIP-seq analysis with our differentially expressed mitochondrial genes. I found that I would say more than 66% of our differentially expressed mitochondrial genes can be possibly regulated by YY1 alone, or by YY1 and C26, or by YY1 alone. And also was shown that YY1 can bind to the promoter of the sirtuin-3, whose role in the mitochondrial functions was discussed earlier. And next we wondered if these changes were observed in the mitochondria-related changes were observed in the knockout mouse brain, if they're relevant to some pathways of neurodegenerative diseases. Therefore we performed the genetic enrichment analysis with several with all the genes changed significantly between the conditions in our RNA-seq data, and constructed an enrichment map of the overrepresented term. And one of the clusters, I would say one of the big clusters in this enriched map was related to various neurodegenerative diseases and all of them were connected to the oxidative phosphorylation. So next we elucidated the core enrichment genes from this analysis for the selected pathways of neurodegenerative diseases, and found that like more than 60% of these core enrichment genes, they are related to mitochondria. And among these pathways, the highest number of mitochondrial genes change the expression in the sirtuin-6 knockout data was for Alzheimer's disease pathway. So confirming the crucial role of sirtuin-6 in the regulation of mitochondria, and in turn in the regulation of pathways, regulation of neurodegenerative pathways. So we, based on our findings, we proposed a model that brain sirtuin-6 knockout mice, it develops mitochondrial dysfunction, and that in turn can lead to the progression of alteration in pathways of some neurodegenerative diseases. These findings were published in the Cell Death and Disease journal. And overall, we found that sirtuin-6 is a key regulator of the transcriptome and metabolome in the brain. We found that the changes induced by the sirtuin-6 deficiency, they are primarily associated with mitochondrial function, and found that the ablation of sirtuin-6 in the brain, the ablation of sirtuin-6, it alters the abundances of several metabolites related to the essential mitochondrial functions like oxidative phosphorylation, for example, and Krebs cycle. And sirtuin-6 can accomplish its mitochondrial regulation via the interaction with two mitochondrial sirtuins, sirtuin-3 and 4. Or also it can cooperate with YY1 protein that in turn is implicated in the pathological agent brain program through the interaction with TP33-IS1 long non-coding RNA. And results that I just presented, they were published in 3 papers. As I mentioned, Cell Death and Disease Metabolized Journal and Aging Journal. one paper was submitted after the thesis was written. I also co-authored a number of additional publications that are not included in the thesis, but anyways, they reflect the number of projects I was involved in. And in the end, if I have time, I would like to thank all my colleagues from both labs for their help during all these years of my PhD. And I would like to especially thank my scientific advisors, Professor Ekaterina Khrameyeva and Professor Deborah Teuber, for their help, patience, and wholehearted support during all these years. I would also thank my PhD committee members who carefully reviewed my work. And I'm also grateful to the education department of the Caltech, who helped me to prepare, who helped me to do all this preparation before the defense. And in the end, I would also thank my family and my wife. Thank you for your attention.
Thank you very much for this very clear presentation and for staying on time. And now we have a question session. So I'm afraid everybody on the committee has to ask at least one question. We can start from people online because maybe it would be just more polite to ask people who are guests to start first. Maybe start with Professor Munoz, if possible. I have to apologize when introducing you. I actually, because in the slide somehow they just had your name but didn't have your last name. Sorry about that. Do you have any question to Dmitrii?
Yes, sir. Don't worry. Actually, my last name is, I have two last names. So Spanish people will have two last names. So my first name is Carlos, and then my two last names, Sebastián Muñoz. So it's fine. Well, first of all, I would like to congratulate Dmitrii because his presentation was very clear, very nice. Also, his written thesis was very clear. Was a real pleasure to read it with a comprehensive introduction. The results were very clearly presented. So it was really nice to read this thesis with very relevant and important results on the sirtuin fields, on the aging field as well. So it was really a pleasure. I would like to extend my congratulations as well to the supervisors. So I'm sure that they supervise very well the PhD candidate, and I can see in some of the results the touch of at least David, who I know personally. So I would like to congratulate for that also. And of course, we always have some questions. So I would like to start first with acknowledging that Dmitrii addressed all my concerns during the revision of her thesis. So I'm not gonna get into those specific concerns because he already addressed them in the revised version. But I would like to ask few things because in the introduction I noticed that in page 27, I mean, there is always things that you have to ask and to point out just to prove that you have read the thesis. So it's what I'm gonna do now. So in page 27, he discussed about he mentioned that the role of YY1 protein in cancer. And he said that there is some controversy because it has some tumor promoting, and so it might act or could act as an oncogene or a tumor suppressor. However, only the oncogenic function is discussed. So I don't know if this was just that he forgot about the tumor suppressor part, or if there was any particular reason for just mentioning the oncogenic part. But I mean, this is just a minor thing that I wanted to point it out. Now, getting into the questions, I'm not a real expert on the analysis of metabolomics and lipidomics and all these omics data sets, but since Dmitrii devoted one full chapter to set up a new analytical tool to analyze lipidomic data, I wanted to ask, just out of curiosity, why is lipidomics more challenging to analyze than any other metabolic or metabolomic data?
Yeah. First of all, thank you for your words, for kind words. And addressing your question, I think it's more complex because I found much less tools, much less published methods for that were developed specifically for lipidomics. For example, I know that some steps in the analysis, like in the processing of raw spectra, they are more or less the same for both lipidomics and metabolomics. However, I think the annotation of lipidomics is more tricky. That first of all, because of most of the lipidomics compounds, they, not the most, but several of them, they may have some almost the same molecular mass, and therefore it's very hard to differentiate between these lipid species during the annotation. So I think for lipidomics, in the analysis of lipidomics, there is a need of the proper tools for the annotations. And also in my personal experience, I know that there are some published tools for that aimed for their analysis of functions of this of the differentially abundant features in metabolomics, but I recall only one tools that were developed for the lipidomics, and in my opinion, it's it's not a comprehensive algorithm, so it yields not so many useful information for the functions that significant lipidomics features are involved in. So I think maybe these two reasons. But yes, as I said, our tool it's applicable not only for the lipidomics. So originally we developed for the lipidomics data because there was a need in the analysis of lipidomics. But I think with some modifications, it can be also applicable with a modification, as I mentioned, in the annotation of the lipid species. It also can be applicable for the metabolomics data. So okay. I think so. At least some of these functions we already applied for the analysis of metabolomics. I can show that. Sorry. One slide where we tried to also, yes, we tried to apply this pipeline with for the analysis of different metabolomics datasets we worked with, derived from the brain CD6 knockout profiles. For example, two of them, the first one and the second one was SHSY 5Y cells, and the last one was from for the brain metabolomics. So some of this function from this pipeline, the developed pipeline, we also applied for the analysis of this data set. However, sometimes they were useful, sometimes they're not, but yeah, still.
Okay, thank you. And did you, why, so you showed that you did metabolomics on the cirrhosis wild type and knockout embryonic stem cells,
Yes.
But didn't you check, or you didn't do lipidomics on those samples, right?
Yes. The lipidomics, it was from the brain samples, mouse brain samples.
But since it didn't work, or you didn't get, I mean, it worked, but you didn't find very significant changes. Why not focusing also on embryonic stem cells, like for the metabolomics?
The reason is that we had the reason is availability of the data. We have several data sets that are unpublished. One of them from HeLa cells. I'm not sure about mouse embryonic stem cells, but we still continue to reproduce these lipidomics results, or at least try to increase the number of the samples to obtain more like reliable results for the sirtuin-6 knockout and volatile samples, because with current results, it's hard to say something for sure about the role of sirtuin-6 in lipidomics. But yes, we didn't focus. We just didn't focus on mouse embryonic stem cells lipidomics, because our main focus was the brain, actually. And the mouse embryonic stem cells, it was chosen because of the high quality of the data set. It worked much better than others. For example, in this slide, I showed two data sets that also worked very well for the SHSOR cells, but it's a different organism and it's cells, also cells. So we decided that mouse embryonic stem cells themselves, they are more relevant to our research and to the paper we published and to the thesis.
And actually it was a nice model, because it recapitulated some of your transcriptomic data. So it's fine. Yeah. Okay, so moving to next chapter, you found that there is a lot of changes in the mitochondrial, or in the genes that encode for mitochondrial proteins, but and you saw also that the mitochondrial masses change. So it is decreased. And then you speculate about the fact that it could be like some changes in the mitochondrial dynamics. So did you have the chance to check that, if there is any change in the expression of proteins that are associated with mitochondrial dynamics, like mitofusins or DRP1, or if there is changes in mitophagy, for example, that could explain these differences in the mitochondrial content?
Yes, we checked it. I personally checked it in the RNA-seq data in the expression of these genes for the mitochondrial fusion, fission, and the mitochondrial also biogenesis related genes. I also checked them, but the changes were like not so consistent, and a lot of genes that were that we analyzed that are related to the mitochondrial dynamics, they were they didn't show any significant changes. So we focused on the lines that has like more clear effect, I would say, regarding the oxidative phosphorylation. So for the mitochondrial biogenesis, we did some, as already showed, we did experimental. We did some experiments, but me personally, I didn't, I wasn't able to extract like very extract very clear differences between wild type and knockout for these genes.
And related to that, so because C6 is also involved in glucose metabolism, so and controls the pyruvate fate. So if in 6 knockout cells, pyruvate cannot go to the mitochondria or go less to the mitochondria. So could affect that, and this could affect mitochondrial metabolism or OXPHOS in general. So do you think, how can you balance, or what is your speculation on what is the major component here, or the major role of 6 here? Controlling transcription of mitochondrial genes, or affecting mitochondrial metabolism that this could impinge on mitochondrial metabolism? So can you speculate a bit about these two things? Because if you don't observe like changes in mitochondrial dynamics, for example, at least at transcriptomic level, maybe functionally you will find something different, but maybe it's metabolism impact, the series control metabolism impacting on the transcriptional level, or in an indirect way, or how can you explain that?
Thank you for the question. Yes, I think it's more transcriptional regulation of the mitochondrial using the genes that encode protein, the mitochondrial proteins. I think that because of the main function of C26 as a histone deacetylase, so I think it can alone or with cooperation with some other proteins like some transcription factors, and can deacetylase the promoters of mitochondrial genes, or deacetylase the promoters of the transcriptional factors that can alter the expression of mitochondrial genes, and therefore also alter the mitochondrial function. However, regarding the glycolysis, we also saw several slides related to the glycolysis story. Yes, in our metabolomics, we also checked metabolomics from mouse embryonic stem cells. We also checked the differentially abundant features and their functional relevance. And yes, we saw some categories, enriched categories. And the top 2 of them was related to glycolysis, Warburg effect, that exactly was shown before us in the but not in the brain. For example, it's similar to the CD6 actioning in the cancer cells, for example, with the activation of anaerobic glycolysis. And yeah, I think also there is a possibility all these changes we saw in measurement of basic mitochondrial parameters like mitochondrial membrane potential or ROS production or mitochondrial mass. It could be a consequence also of the enhanced glycolysis, because we know that in some tumors or enhanced glycolysis, we saw the hyperpolarization of the membrane. So it's this condition also decreases the mitochondrial membrane potential, and I saw some papers that stating that all these parameters that we also measured, like increased ROS production and increased mitochondrial mass, it's also common for the cancer cells. So maybe yes, maybe it's also a consequence of the enhanced glycolysis in the brain. However, I would be very accurate to say that all these changes are due to the enhanced glycolysis. So I think all pathways, all the axes that can C26.
I agree. I mean, you see clear changes at the transcriptomic level. So I do really believe that both transcriptional regulation and metabolic regulation can coexist and can explain your parts or different parts of your phenotype. Yeah, absolutely. Naturally, I like that you put this slide now, because you are kind of anticipating my next question. So you clearly see a decrease in membrane potential but on the other hand, you see increased ROS levels.
Sorry,
that you see decreased mitochondrial membrane potential, but at the same time, you see increased production of ROS levels from the mitochondria. And this seems counterintuitive because normally by decreasing mitochondrial metabolism, not always, but most of the times by decreasing mitochondrial metabolism, what you achieve is a decrease in ROS levels. So what you see here is more like a mitochondrial dysfunction in general, or how can you put together these two findings, the decrease in membrane potential and also an increase in ROS levels?
Yes, as I said, it's the increased. Yeah, let's start with it. This analysis with these experiments were done with different types of cells. So it was human brain cells, SHSY, 5Y. So we used for some other metabolomics experiments, and our primary metabolomics data set was from mouse embryonic stem cells. So it's a little bit different cells. However, I think as I already mentioned, all these parts, they've already shown for the enhanced, they were already shown for as a consequence of the enhanced glycolysis. So let me return to the slide with a paper, with the two papers that were originally described the role of student 6 in the mitochondrial regulation and glycolysis. So here we also, in one of these papers, we saw the downregulation of the mitochondrial metabolites. And most of them, maybe not most of, but some of them, I truly believe they are the same as we see in our data set. So I think it fits. And I remember that in this paper, there was also shown the decreased level of the ATP production. So I don't think it's a controversial story. However, yeah, say it for sure, we need to like repeat the experiment with metabolomics, or not to repeat, like analyze metabolomics from the brain to say that to accurately show that these metabolites that we already shown in mouse embryonic stem cells, they are decreased in student 6 deficient mouse brain as well, not only in cells.
I mean, my point was, I mean, it could be possible. I mean, mitochondrial dysfunction can be associated with either increased ROS levels or decreased ROS levels. This is fine. It's not in contradiction with anything. My point was if in your data set you find any gene pathway or any gene associated with ROS production or ROS detoxification that is changed, that could explain why you see those changes in ROS. I mean, it's not that the data is not solid, because it could be. It's just if you have any evidence to support that, or to explain that the ROS levels are increased.
Yeah, I got your question. Thank you. Yeah, I think there are some. I checked the genes in RNA-seq data. Several genes that could be also involved in this process, but I'm not sure about their changes. I don't think the reason why I didn't show this, I think it's because most of the changes were not regarding this increase, like ROS production and detoxification. There was not so abundant like changes for oxidative phosphorylation. So maybe I saw 2 like of 3 genes related to the antioxidant parts in our data, but I remember that for example, in the mitochondrial pathways, in the notation of mitochondrial pathways I used, there were much more antioxidant genes. So it's 2 or 3 genes. It's not enough for me to state, to claim.
Okay, it's fine, yeah. Okay, so another question I have in this chapter is the crosstalk between, or when you try to dig into the molecular mechanism, you see crosstalk between CIRCs and other mitochondrial sirtuins expression, and you find that the YY1 is one of the molecular players in this mechanism. And you found that YY1 is sitting on the promoter of C3 and or specifically C3, probably also on C4. Is CIRCs already or also sitting on the promoter of C3 together with YY1? Have you checked that in your ChIP-seq data?
Yeah, I checked. It's not in this ChIP-seq data at least, but maybe not in this ChIP-seq data. But there were a paper showing the axis for the regulation between CERT6 and CERT3 through the transcription factor of NRF2, as far as I remember, as far as I recall. And in other ways, they can interact with CERT6 and CERT3. It was described in the string network. So we also analyzed the interactome in top proteins that interact with sirtuin-6, and maybe all the mitochondrial sirtuin-6, mitochondrial sirtuins was there. So that was the reason we also say that not one of the reason, but it's an additional evidence that 3 and 6 can interact.
Okay, yeah. Okay, I mean, I have more questions, but maybe I should leave my colleagues the opportunity to ask things. So again, I would like to congratulate you again for your very nice presentation and also your very good defense now. And yeah, so for my part, I'm done. Thank you.
Okay, thank you very much for these questions and sparking this discussion, and actually giving Dmitrii opportunity to show many more results than he had during his main presentation. So we saw he did actually much more. We can return to you, Professor Munoz, for the second round of questions. And now let us talk to another online participant, if we have online, Professor Peshkin, Leonid Peshkin. Do we have?
Yes, I'm here. Can you hear me?
Yes, can see you and hear you. Thank you very much for participating in this. And if you have any questions, please ask.
Thank you very much for including me. I think Carlos largely reflected sentiment of many of us. It was a great body of work. It was very nice to be part of that. As far as questions and feedback, of course, I provided some of the questions and feedback already, and it's been nicely taken into account. So at this point, I only want to ask a general question. Always when you complete such a substantial body of work, you feel you could go back and do something different. So I'm curious to ask, what would you have done different? And also, if it's not time that you were limited by, what kind of technical limitations you experienced? So maybe you feel that if lipidomics had much nicer resolution or identified more species, you could have answered many questions better. Maybe you feel like you're missing some substantial data set, which would be a crucial piece in investigation. Maybe you would have designed some experiments if you could have time and resources to start some measurement from scratch now, that would have been really helpful. This sort of thing, if you could comment, you know, somewhat technically what you were missing and what you have.
Yeah, thank you for the interesting question. I think back to the start of the project, I would we originally wanted to do the truly multi-omics integration of the data. So my main maybe it's not a sadness, it's just a limitation of the work that I would have a multi-omics profiles from the same organisms, from the same biological materials, maybe from cells, maybe from brains, but to achieve some real multi-omics integration, it would be much interesting. However, I think I like what is done. So back to the start of the project. Yes, I would analyze their profiles from one from the same tissues from to the real multi-omics. So that would be great.
So maybe just a short follow-up in terms of data that you did see. If you could have gotten better technology to work for you, you know, deeper measurements in terms of gene expression. If you wish there was a better instrument somewhere, what would it be? Where I get it, multi-omics, but perhaps another idea of where the present technology protocols, instruments limit what you could have done.
RNA sequencing, yes, I think I forgot that one of my dreams is to analyze single cell RNA-seq data of CD26 knockout and wild type mice. So it will be super cool for for me to do such analysis. So maybe this is a method, this is an improved method you mentioned. Maybe it's not so improved, it's just more it's a special data, so it will be a little bit more informative than results from the bulk RNA-seq from mix of cells. So the specification, the differences between cell types, it will be also great for my work.
All right, thank you. Yeah, I think if we could probably, if we were not forced to reason about activity of pathways from transcriptomics data, but had a better view at protein abundance and protein modifications, more of that kind of insight, that also would have been very helpful. But thank you very much. I don't think I have more questions. Again, thank you for including me, and congratulations on excellent body of work.
Thank you very much. Okay, then let's move to another online member, to Professor Leprevier, Gabriel Leprevier. Professor Leprevier, are you there?
Yes. Can you hear me?
Yes, can hear. Please speak up a little bit. We have a little bit of a problem here of the dynamic system. So.
Okay. So thank you for inviting me to review this thesis. I have to say that it was a very well written thesis, very structured, very clear. I really enjoy reading it. It was really written like very concise and to the point. There were a lot of very interesting data, and especially about CERT6. And I was very intrigued with the connection that was brought between transcription, metabolism, aging, and cancer. Especially that I have a lot of interest in transcriptional regulation and cancer, and this brought a very interesting angle on that. So I have a few questions that will be more like about some of the results and try to speculate about some of the findings. So first of all, I found it quite fascinating that CERT6 can both repress transcription and activate transcription. So how do you see that, and how do you think CERT6 can both have this activity, and what will drive that? What will be the context when it will repress or it will activate?
We cannot hear anything, maybe.
Yeah, I don't hear anything.
Yeah.
So.
Okay.
Neither can I.
Okay.
Yeah. So I'll repeat. CERT6 is a histone deacetylase, so it's repress the transcription, but I think it can achieve the regulation of other proteins with mono-ADP ribosylation, and through which it can transcriptionally repress or activate other genes. So directly, 36, I think it's a transcriptional repressor, but the activation of the activity of other genes is possible through the interaction with some transcriptional activator or repressors like YY1, as we mentioned in the thesis.
Yes. So then my follow-up question is, what will mediate the specificity of action of CERT6 on specific genes?
Sorry, could you repeat again? I can't hear you.
So the thing is, you show, for instance, that CERT6 will activate the transcription of a number of mitochondrial genes.
Mm-hmm.
So there is specificity because it's not activating all the genes, or it's not repressing all the genes. So can you tell us more about how it's how this specificity is mediated?
Okay, I get the question. I think the specificity it's due to because of their specific sites. If we're talking about the direct regulation of the mitochondrial genes, I think it has a specificity in sites to the deacetylation. So it's it can only deacetylase the subset of the several promoters of the promoter of cellular genes. Mm-hmm. But I cannot say for sure. I think it also has specificity in mono-ADP ribosylation for other proteins, other, for example, transcription factors that can in turn activate or repress mitochondrial related genes. So I think if speak of the direct regulation, it's due to because of the specificity of this histone deacetylation sites.
Okay. So you show that there's a strong co-expression between CERT6 and CERT4 or CERT5 in brain, and I found the R values very high. So it's a very tight co-expression. So do you know what explains that? Are they directly co-regulated, these genes, or?
I think it's we didn't check the molecular aspects of the regulation between CYP6 and mitochondrial sirtuins. So because I'm as a bioinformatician, I a little bit limited in methods as I can apply for the to clarify the mechanisms of interactions between the genes or between the proteins. So I worked with the data available. So well, as I said, we didn't check the molecular aspects, but we do check. We do check, and thank you again for to Ekaterina Yeremenko, who checked, who did an experiment with the recovering of this mitochondrial of the expression of the genes encoding mitochondrial sirtuins 3 and 4. So in sirtuin 6 knockout cells, the overexpression of this SIRT3 and SIRT4 in the sirtuin 6 knockout cells, I said it may it may rescue the mitochondrial membrane potential. So this is one validation for the crosstalk between SIRT6 and SIRT3 and 4, but yes, again, as I said, we didn't check the molecular aspects of the interaction between SIRT6 and mitochondrial sirtuins.
Yeah, I understand. I think it's something that can be very interesting for some other labs to find out. So I want to try to clarify about the effect of SIRT6 on glycolysis, because what you mentioned just before, that what is published is that the SIRT6 knockout have a higher glycolysis. But in your data, I see that a lot of glycolytic intermediates are decreased in the SIRT6 knockout. So how do you put this together?
You mean the metabolomics data, right?
Yeah, what you generated. There are quite few glycolytic intermediates that are decreased in the knockout. So how do you put this together?
First of all, I didn't show all the differentially abundant metabolites in this slide. Okay. So there are more. It's just a subset to show the differentially changed, statistically significant metabolites, mostly related to the oxidative phosphorylation and the Krebs cycle. But speaking about the glycolytic genes, they can not genes like metabolites. We didn't check. I already showed the enrichment of the these significant metabolites, and that we have glycolysis as enriched categories, but we didn't check the direction. So I'm not sure. Maybe
no, it's just based on.
I also saw several metabolites that are downregulated. However, it's I'm not sure that for the full set of metabolites, they are all downregulated or upregulated. So,
but for instance, do you know about pyruvate or lactate?
Sorry.
Do you know what is the level of pyruvate or lactate in your data?
I didn't see lactate. Okay. Yes, yes, I didn't see it in the slide, and I'm not sure that we measured it. I need to check it, so I cannot say it right now. But again, we just we didn't focused much on glycolysis in this data. So.
Yeah, I understand, of course.
However, I understand that this might be also possible mechanisms of why we see these changes.
Yes. No, actually, there's something that was striking for me is that the phenotype you see with mitochondria, that it's less active and in the CERT6 knockout. For me, because I work on the response to glucose starvation, it's actually suggesting me that the CERT6 knockout cells may be more sensitive to glucose deprivation. And can you tell me if there's anything known about that or what is known about that?
The phenotype, yes, I think regarding the implications, the transcription of regarding the implication of C26 in the mitochondrial, this in part mitochondrial phenotype, there was also a paper. There's also a paper. I think I have another slide for that. It was yes. Here it is. It's for the not for the brain, but it's data derived from myofibers, but also from mice. They actually have the same. So it's this showed the same more or less the same changes, but not in the brain. They also show in part mitochondrial biogenesis, oxidative phosphorylation, all that we see in the we see in part in mitochondria in our data. They also performed several experiments with oxidative phosphorylation coupler and several transport chain complex inhibitors. But they propose different axes like STRN6. It can act through the transcriptional factor CREB to regulate the expression of SOC6 and then affect the mitochondrial function. But in our data, we didn't saw these changes that were presented by that were presented in this paper. So we decided that in the brains it's different, and therefore we focused on the other axis possible regulation. But speaking about what's known, yes, the impairment of mitochondria in more or less the same genes. It was already not already, but it was shown more or less in the same time, and the same in the time that we submitted our paper on the review. But I think it's consistent with our data.
Yeah, that's very good to see. Of course, it's confirmation. But actually, my question was more if there's anything known about how CERT6 knockouts respond to glucose deprivation, because my prediction is that based on this phenotype, they may be more sensitive.
CERT6 knockout glucose deprivation. I think CERT6 can regulate transcriptionally the expression of the glycolytic genes, thereby affecting the glucose metabolism and glucose deprivation. So as far as I remember, it was it was shown in one of the papers I already mentioned today. So, yes, I think through this axis.
Okay. So I would just ask a last question for now regarding the CERT6 and aging. And so I understand that the expression of CERT6 is decreasing over lifespan. So do you know what is causing this decrease, or is it known?
Could you repeat the last part of the question? Because
yeah, what
maybe
do you know? Do you know
a little bit louder? Because
do you know what is causing, or is it known what is causing the decrease of CERT6 expression during aging?
coding CERT6 expression, right?
Yes.
So the question was about what was the cause of decrease of the reason why CERT6 decreases expression in aging.
Yes, I think it's one of the reasons is because some of the because of the dysregulation by one of the possible ways, the dysregulation of transcriptional factors that can activate, that can regulate CERT6 activity, and also it's because of the, I think the NAD plus levels, it also dramatically decreases with age, and NAD plus is required for the activity of sirtuins. So the way and I know that some possible activator of sirtuins or we tried to develop to activate sirtuins, and they work in it's more or less the same way to compete with the nicotinamide for the binding sites on sirtuin 6 to activate its expression in aging. But again, I think there may be different ways of the dysregulation of sirtuin 6 through aging.
Okay, I think that makes sense. I thank you. I will stop for now, and I will leave it to the other jury members.
Thank you very much for your questions. And now let's move to one more online member, to Professor Pervushin. Professor Pervushin, please, if you have questions.
Hello, can you hear me?
Yes.
Oh, okay. So let me, as previous speakers, I would like to acknowledge the large body of work Dmitrii has presented. This is truly multi-omics integrative study, and the amount of work is really, really large. It's really nice and to see works like this to be done by PhD students. So my congratulations with this big and successful work. Many of my questions have been asked already by other jury members, and my criticism towards Dmitrii's work was mostly about the literature part, and all that has been addressed in the revised version. So Dmitrii reworked the abstract and the introduction. They are quite nice, like little short, but still they are on their place, and I think I'm satisfied with all my comments that I had before. And for now, I think the question of correlation of CERT6 and CERT4 and CERT5, I just want to ask out of curiosity, the functional studies, for example, knockouts, are they phenotypically different if you knock out CERT6 and say CERT4?
Yes, they phenotypically different in different tissues, because CERT3 and CERT4, it has mitochondrial sirtuins, they have functions primarily related to the mitochondria. So most of their functions are related to mitochondria, and sirtuin 6 in some studies, for example, in our results, it really shows a connection with mitochondria, but in many others, it's also shown to be mostly involved in DNA repair. DNA repair to facilitate it. It was shown to regulate some, for example, lipid metabolism.
I'm referring more to the phenotypic. So in these knockout models, in these animal models where you knock out a gene.
Phenotypically, yeah, phenotypically, I believe I'm not sure about tissue-specific models, but I believe that sirtuin's full knockout of sirtuin 3 it will also kill the mice very quickly. So I think it's also very dramatic, the loss of these proteins in mice. But regarding the tissue-specific models, I'm not sure. I'm not sure that many studies were published about the CPN3 knockout in the brain, because I found only some separate evidences from different studies, but not but yes, addressing your question in I think full knockout of these genes, they will have more or less the same consequence for mice.
All right, so they don't compensate for each other, right?
I think they cannot fully compensate each other because of the different cell localization, different functions. But say already shown in our experiment with cells, the overexpression of one of these genes and increased levels of proteins of one of these proteins, it can improve the situation. For example, at least for the mitochondria, for mitochondrial membrane potential. But I do not think it will rescue the whole phenotype.
I think that's fine. So I have one more question regarding the last part about CERT6 regulating YY1, which in turn regulates TP73 antisense 1. So my question is, this long non-coding RNA, TP73 antisense 1, it's a long non-coding RNA, right?
Right.
So how is it different? So can you name for us, this is just a general knowledge question, can you name a few long non-coding RNAs that are important for human disease, the ones that you know, and how is this TP73 antisense 1 different from them?
Sorry, regarding the role of TP73 AS1 in aging, I'm not sure about the role of I cannot maybe right now I cannot recall the names of other long non-coding RNAs that are also implicated the regulation you mentioned, but I just wanted to mention that in this work regarding the last in the last part of my thesis, in the last chapter, we focused more, at least me, I focused more on YY1 role, since it's more relevant to my thesis. And the long coding RNAs in biology, it's a big field, but my thesis it's mostly about YY1. In this study, we focused only one specific long non-coding RNA. So yes, I do not have, I think, much.
Okay, so I guess my question still was that, as suggested by the name, it's an antisense transcript to the TP73 gene, right?
Yes.
Did you check if the levels of that, I guess it's a pro-oncogene, if they are also perturbed in this cancer?
You mean TP73? It's a neighbor gene. It's a neighbor gene. They have an overlap in 200 kilobases, but in one of the papers that were published 4 years ago, it was shown that apparently there was no consistent data, there wasn't consistent results regarding correlation of them. So apparently, I know that many long non-coding RNAs are known to be regulate neighbor genes. However, apparently, also have a slide for that. Yes. Sorry, let me check. I think it's not the case for, yes, here it is. And here is a small overlap of TP73 and TP73-1. It's small, but according to the results from the knockdown of, according to the measurements of expression in two cell lines, the results are not so consistent. If we knock down one of the genes, for example, TP73-1, it won't be the neighbor gene won't be changed consistently into data sets. So the authors came to the conclusion that apparently this long non-coding RNA, it's not it could not be a direct regulator of TP73.
Okay, I think that's a pretty good answer. Yeah. All right, so I'm done with my questions and I pass the mic to the next.
Okay, thank you very much, Professor Pervushin. And now let us move to the in-house jury members. Let's start with Professor Dmitrii.
Okay, thank you very much for this nice study and really nice talk and very interesting discussion. And I had actually a number of concerns, but most of them have been fully addressed in the report. And that's why I do not think that we should discuss these points again. But one of these points was about retrotransposons, line 1. And this point was not addressed in the presentation. And so I just remind you this question. The question was related to the story raised by Vera Grabunova laboratory from the US. And Vera showed, with colleagues, showed several years ago that mice deficient for sirtuin-6 overexpressed a number of retrotransposons, and this had a dramatic consequence for the phenotype of these mice. And I wondered whether you, could you address this issue? I mean, did you try to analyze the abundance of, for example, transcripts of retrotransposons and so on? I understand that this is probably not very simple because these are not unique genes, but nevertheless, could you comment on that? Thanks.
Yeah, thank you for the question. Indeed, we did an analysis, and it was one of our maybe first analysis with RNA-seq data. So we obtain, we process the samples, and then we also downloaded the annotation of retrotransposon from the repeat masker website to check the abundance of mobile elements in the genome, mobile elements in our data. And while we had an annotation for different classes of retrotransposons, as you can see in the slide, only few of them showed consistent changes in differential expression analysis of these retrotransposable elements. And regarding the L1 retrotransposons that were discussed in Vera Garbunova paper, we found no significant, no upregulation, not a single retrotransposon upregulated in our data in sirtuin-6 knockout. Some of them were downregulated, but overall the median of the expression changes were near zero, as it was expected. However, it was not, there was no clear pattern of upregulation of line ones. And then we were curious why so. So together with our former master student, Diana Zagerova, we decided to analyze more publicly available RNA-seq data sets and check what is going on with the transcriptional, with the retrotransposon expression in these public data sets. So here's the results from mouse embryonic fibroblast, embryonic stem cells, also from thymus. And if the changes, for example, for long terminal repeats or signs, they're more or less consistent. This graph reflects only the statistically significant retrotransposons. And if for the LTR and signs, the data is more or less, results more or less consistent. For the lines, it inconsistent at all. For example, in mouse embryonic fibroblasts, there was no differentially expressed lines at all. In embryonic stem cells, they were, some of them indeed were upregulated, and most of them, most of the statistically significant retrotransposons were upregulated, while in thymus, they were mostly mostly downregulated, that contradict to the findings from the Vera Garbunova paper. So we decided to leave this line of analysis and focus on the changes that we observe in that we observe for sure. I mean, the mitochondrial related changes.
Okay, thank you very much. It is quite interesting and intriguing. Okay, so I actually had a number of minor issues, minor points, and I would like not to raise these points once again, but now we just, after the talk, I decided to ask one more question. I actually love the story of friendship between systems biology and molecular biology. I mean, in functional terms, that in this study you combined systems biology analysis and functional consequences of your findings. But what about differential expressed genes? So, for example, you found a number of, actually many, of course, many differentially expressed genes. But, for example, in the figure 4 and supplementary figure S1, you shows that you show, for example, 5 genes with a dramatical changes in expression levels. And could you comment on some probably functional consequences of these particular changes? I mean, whether some of these 5 genes, which are the most dramatically changed expression you found, do you think that some of these genes brought some functional consequences, some particular consequences on these brains and so on?
Yeah, thank you for the question. I think for the you mentioned the 5 genes in the figure S1. Yes, first of all, I would like to say that the part of, I think, C4b, CHR, and A1, they were already shown in the analysis that was done before by my colleagues with the microarray data from the brain 36 knockout RNA, microarray experiments of the brain 36 knockout gene expression profiles. But yes, we studied them. I searched for their function and Google them, separately Google them. I think the most relevant of them was SH RNA1 because I saw several papers with its relation to the brain, but we didn't focus on their on these specific genes in our paper because they're all parts of some big pathways and changes in one gene. So it's they're not big and very influenceable transcription factors. So we didn't focus and focus on the relevance to the brain separately, and preferred to discuss them all the changes in the context of mitochondria. But for example, for ZRNAB3, we also showed it. Shows a study showing its relevance to the neurons, and we saw several for this gene. We saw several alternative splicing events. So maybe we will this particular genes. Maybe it will be one of the will be a central part of the another story that we are working on. So.
Okay, thank you very much. I think that is all.
Thank you very much for your questions. And now to Professor Dmitrii Ivankov, please.
Yes, thank you very much for the presentation. And I have only short questions. Actually, after reading the thesis, I didn't have any questions. However, after writing my review and after reading the reviews of other reviewers, now I found one question very interesting to me. I don't remember who was the author of the question. However, I think it is worthwhile to discuss it right now here. And so the question was, if I'm not mistaken, was what is the linkage between the protocol you proposed in the first results chapter to other 2 chapters, and did you apply it successfully to some data? This is the first question.
The linkage is yes, we used it for the metabolomics and lipidomics analysis I showed today. Yes. So here we applied several functions of this pipeline to the analysis, starting from the annotation. So
sorry, I have to leave. We have an alarm.
Okay. So yes, we applied it to the lipidomics data as shown, and for the metabolomics data as well. Let me find it. Yes, and for the metabolomics data as well. But in this data set, we applied only functions from the downstream analysis for the downstream analysis, because this data came already processed from raw spectra, and we already had a table of metabolite abundances. So we applied functions from this pipeline to the downstream analysis of these samples.
Okay, thank you.
Statistical significance and for the visualization, like this PCA plot was also.
Okay, thank you for the answer. And the next question is slide around 33 or something like that, where you showed the correlation between the expressions of between the levels of CERT6 and YY1.
Mm-hmm.
So out of here out of 37, right?
Mm-hmm. Yes, that's 37.
37. Okay, and so out of curiosity is did you check this correlation with other proteins?
I believe this this particular figure was plotted in the response to the reviewers that were asked us about this the correlation between YY1 and TP73 IS1, and in the figure I think we also did the correlation. We also, as far as I remember, we plotted the correlation between TP73 IS1 and TP73. However, the correlation between TP73 IS1 and TP73, it was not me who plotted the figure. So therefore, I didn't include it in the presentation, and I didn't mention it. So my work was related to this. To study the correlation between YY1 and TP73S1, and also to investigate the changes in TP73S1 activity in aging, in Alzheimer's disease, in different tissues, different brain compartments.
Okay, thank you very much.
Okay, so now only me left. I have only a short technical question about lipids. As you showed on slide 19, you didn't see any significant differences, but there still were some differences in lipid composition between brains of knockout mice and wild-type mice. And because later you start to talk about mitochondria, and there are actually some lipid classes which are known to be associated with mitochondrial membranes more than with other membranes. Did you have a chance then already kind of post hoc to examine if these particular lipid classes were changed more than other lipids?
No, we didn't do this. We didn't do it, but we. I would expect first of all the changes in triacylglycerides, because first of all it was already shown in other papers that triacylglycerides are enhanced in sirtuin-6 knockout. It's mostly because of the depleted beta oxidation. Beta oxidation can be also mitochondrial. So if I would do it if we do it right now, I would investigate it in this way. So trying to connect triacylglycerides with mitochondrial function. And also I will try to investigate more carefully, I would say, or more detailed investigation of the different triacylglyceride species. So for example, distribution of their changes in regard of the chain lengths, so it will be also interesting. But answering the question, no, we didn't do it after we presented, after we published the paper, but later I think we will.
Okay. Oh, that's very convincing, very good. Are there any more questions from the jury members? Because maybe some questions were not addressed or some new one appeared.
I've got a little technical question on this particular figure. I mean, the right one. Could you comment on why these two replicas from sirtuin knockout mice are so distantly related to each other? I mean, the very left and the very right rows. Yeah. Okay. Thanks.
That was one of the issues we encountered when analyzing the data set, and maybe it was one of the main limitations that didn't allow us to find any significant changes. So why so? It could be also because the normalization of these samples was different. So maybe it's due to the incorrect normalization, but since we didn't have internal standards, or we didn't have we also we had only blank samples for these experiments, as far as I remember. So we tried to normalize by them, but maybe it was not enough, and that's why we have what we have. So I know that there was another data set for lipidomics generated. I also mentioned HeLa cell data set, but for this one we gave up also because of their difficulties with normalization and with small sample sizes, with all these limitations that
okay, yeah, thank you very much.
This analysis
I think
incomplete.
It is important for us that this is certainly not because of your pipeline, correct?
Yes, yeah, sure. But as I mentioned, we worked with I work with what I have. So it's I can say that for this data set, I tried really different possible normalizations. So this one was normalized with assumption more or less simple, like median normalization, in addition to the blank with a subtraction of blanks. But we really tried different methods, and this was they resulted. None of them resulted in the proper clusterization. So
okay, thank you very much.
Okay, are there any more questions from the jury members? If there are none, are there any questions from the audience? We always encourage the audience members, students, postdocs to ask questions. Okay, nobody there to ask questions. All right, it was a very productive, I think, Q&A session. Thank you everybody for being so active. It gave us opportunity to go much deeper into results and to see many more results and analysis done during this actually PhD work. And now, moving from the Q&A section, we go to supervisors. So I can see Professor Teuber being back, great. And if you could say a few words about Sergey and his work.
Yes, I think it has been a pleasure to work with Dima. In addition to all the things that you have seen, he has several more projects that he's helping us and collaborating with us to analyze a lot of data. And he always comes up with ideas and troubleshooting for different methodologies, but also ideas to analyze or compare with many other data sets. So it's like an ideal student that also is a hardworking student and also has ideas and also reads the literature. So I am really proud of him. So Dima, congratulations on your work. And although I am a cell biologist and molecular biologist and really needed confirmations, I really appreciate the work and how difficult it is to do bioinformatic analysis through our work together and with Katya. So thank you for being my student during this time. And it has been my pleasure, and I wish you all the best for your career in the future.
Thank you very much, Professor Teuber. And now the main supervisor, Professor Kremeva.
I totally agree with Debbie that it was a great pleasure to work with Dima, and I can only add that for Dima, this PhD was more difficult than for average PhD student, because firstly, of this joint program, yeah, some formal things Dima had to do twice, like writing some reports twice, taking courses twice, so it meant more workload for him. And also because of having two supervisors, I think it was more difficult for Dima, because sometimes I was asking Dima to do something, to participate in some project that was interesting for me. And sometimes Debbie was asking to do him also something that was interesting for her, that in the end meant not twice, but definitely more work for Dima. And as you have seen from his answers on questions, what he included in the thesis maybe was only half of what he really did, and large part of his work is still either under review or in papers that we have in preparation. So maybe if we waited for 1 year or so until all these papers are out, Dima would have something like, I don't know, 8 papers to defend with, but we decided that we should not wait because already right now he has definitely enough to pass all the formal criteria to graduate with great thesis. And also because I want to say why it was difficult for Dima, because he comes from physics, right? He has physics background, and he did not receive neither formal training in bioinformatics nor in molecular biology. But right now, as you see again from the work he has done and from the questions, how he answered them, he's qualified both in bioinformatics and when I say he is qualified, I really mean it, because again, because of this extra workload we put on Dima, it ended well for him actually, because he did have a chance to work with every possible data type. I can imagine he worked with RNA-seq, with ATAC-seq, with ChIP-seq, with Hi-C, with lipidomics, and with metabolomics. So professionally saying, I cannot think about anything else Dima could get experience with during this PhD. And also for bioinformatics students, at least in my experience, it's typical that they usually focus on the technical part of the data analysis and leave interpretation of the results to the wet lab colleagues. But Dima is not the case. He really looked deeply into the biology part of his studies. And I think this is thanks to Debbie, because she taught him this molecular biology part of his during his PhD studies. So Dima, now I can surely say that he is equally qualified in the data analysis part and in the wet lab part, and which makes him a great specialist and great expert. And I can only wish him luck in his future career and his future endeavors in academia. And again, it was a great pleasure to work with you, Dima. Thank you.
All right, great. Thank you very much. And now we have a formal part with a closed door deliberation. So unfortunately, I have to ask the supervisors and all the visitors and everybody except the jury members to, for those of you online, there will be a special link provided. Maybe Elena can help me.
Yeah, sure, with great pleasure. There will be no separate link. I will open a breakout room only for the jury members now. Professor Toiber, you can stay online. Just stay in the main session. Once the jury is ready with the decision, we will be back for the announcement of the result.
So welcome back, everyone, and I'm happy to announce a unanimous decision, actually quite rare for such a big jury, to accept thesis as is. It means everybody was completely satisfied with work, with changes we made, and especially with discussion. It was really a great presentation, great job, and I think I'm speaking for everyone here, and there were comments during the deliberation of what you've done, really good job, both writing thesis, doing your work, of course, and answering the questions. Thank you very much, and congratulations to you and to your supervisors. Thank you.
Again, thank you, everyone, for coming. It was really important for me to have you all there, or have you all here. And I would like to additionally thank the jury members that thoroughly reviewed my thesis, that asked me a lot, that I would like to ask you for the deep understanding of the work, of its limitation, of its maybe some good aspect of my work, and I really appreciate that. Thank you very much, and thank you again, my supervisors, because without you, it was not possible to do all this job. Thank you again.